Fine. All right, pay attention now. So coming to our revision, quick revision. So what we discussed, we already know what are stem cells, difference between stem and normal cells, or not adult, normal cells. Then we discussed the properties. What are the three properties? Differentiation. Three properties of stem cells. Renewability, potency, plasticity. Okay, very good. See, conversion of stem cell to the other thing is called as differentiation. Then we discussed about potency. There are 4 types of potency. So we have totipotency, pluripotency, multipotency, and unipotency. So totipotency, they are actually the sperm cells and ovum. Okay, the zygote actually. Okay. Pluripotent also a zygote actually. In the zygote, the embryonic cells. Embryonic cells will be pluripotent cells. They can make almost all cells of the body. Whereas in multipotent, now they will become 3 important lineages. Just a simple flowchart. We have 3 types of germ layers in a zygote. I will show you the germ layers. See here. We have ectoderm, mesoderm, and endoderm. So these are the 3 germ layers, and these are various cells originated from these germ layers. We have neural and epithelium. Keep in mind, interesting here, both neural cell and skin cell are same. Technically speaking, we can convert a skin cell back to a neural cell because they both have same origin. They have same lineage. Okay. Then we have mesoderm. From mesoderm we have red blood cells and cardiomyocytes or cardiac cells. Then from endoderm we have liver cells, pancreas, and heart cells. Alright. So these are the various types of cells. Then, even more depth if you go. These are the mesoderm, ectoderm, and endoderm. This is a placenta and this is a baby. So inside the growing baby, apart from yolk sac, these are the 3 germ layers and the various cells originate from those germ layers. Okay. So see here. So technically speaking, even a hair can be converted back to a nervous cell. Technically, because they all sharing same germ layer. Okay. So that is really surprising. Just to take a hair cell and you can convert it to a brain cell. Simple. Because they have same roots. Okay. That we'll see. Next one. Next we discussed same concept. Then the most fascinating thing I felt was this one. See here. There is no much difference between the embryos of various animals. See, the embryo of a shark, lizard, chicken, pig, and human have almost same developmental stages. If you see there, this one. So, see, almost everything looks same. The reason was very simple. If you see the evolutionary tree, all animals sharing a common ancestor at one point. So from that common ancestor we slightly deviated at the top of the tree actually. But our roots are same. So the genes the makeup of all mammals or all animals, land animals, have sharing same genes. That was very prominently visible in the embryos actually, embryonic development. So in order to understand the stem cells, first we should understand how the embryo develops. Because it is the starting point, that is the stem cell that is taking various lineages to become various cells. So embryology and stem cell technology has a deep connection to understand the various lineages of various cells, even to evolutionary point of view. This picture was drawn by Charles Darwin 200 years ago. Even Charles Darwin was fascinated by seeing similarities between the animals. This is the same reason why humans can able to survive in aquatic environment for 9 months. Why we are surviving in the water, in the amniotic sac? The reason is we have those genes, we have those cells, the cells that can sustain us in an aquatic environment. That's very interesting. Then coming to the types of stem cells. So broadly, the stem cells are, let me write here, multipotent are ectoderm derived, mesoderm derived, and endoderm derived. That's it, based on the potency. Then next we discuss about types of stem cells. types of stem cells we discussed. There are three types of stem cells. What are those? Embryonic stem cells, adult stem cells, and induced. I was small letter actually. Induced pluripotent stem cells. So, what is the difference between induced pluripotent stem cells from other cells? So, keep in mind, induced pluripotent stem cell means we have taken an adult cell, adult matured cell. Don't confuse with adult stem cell. I am not discussing about adult stem cell. I am discussing a terminally differentiated cell. Oh, this is a new term to you. Terminally differentiated means that is the final stage of the transformation from stem cell to terminally differentiated. From stem cell to a hair, stem cell to a skin cell, stem cell to a mucosal cell. These cells are terminally differentiated cells. Got it? Matured cells are called as terminally differentiated cells. Now, the hypothesis was Shinya Anamaka. There is a scientist called Shinya Anamaka. The greatest scientist actually. He is a Korean, I guess. So what Shinya Anamaka did was, he understood that Dolly experiment, you remember? So we have a worm. In the worm we have a niche. N-E-C-H-E, niche. So niche means it has some environment, some chemicals are there. These chemicals activating the genes and those genes started replicating the cells. Clear? Now, he identified 4 important transcription factors. There are 4 important transcription factors. Actually, there are almost 200 factors. They found 200 factors, but they want to know what exact chemical composition out of 200. Maybe I can say like this: among these 30 students, 3 students know how to make a computer. My goal is to make a computer, and I want to filter out of 30. So he was doing this research for next 15 years. He was trying with various combinations. He maybe one person here and here. So first he took this group, he didn't get the results, then this group, then combined group. So he is working with various types of chemicals. At the end of the day, one day he got a result. He came to know that there are 4 important transcription factors. Those transcription factors are c-MYC, OCT4 and SOX2. There are some scientific terms. OCT4, SOX2, c-MYC was not here, but c-MYC was C-MYC and nano-G. Okay. So what he did was very simple. Now, if these chemicals going to activate the chromosomes, if these 4 chemicals going to activate all the chromosomes, got my point? The hypothesis is all 46 chromosomes can be activated by 46 chromosomes activated by these 4 chemicals. 4 chemicals, right? So he took these 4 chemicals in a test tube and he took a normal cell, a normal terminally differentiated mature cell, or in a nutshell, he plugged his hair and he suspended his hair in this chemical. You know what happened now? In the hair bulb, there is a cell, right? That cell slowly went back to a stem cell stage. The other cell became a stem cell. You know what is the reason? All 46 chromosomes activated. Now this cell can able to transform to any type of cell. Now it became an embryonic cell. So another cell became a stem cell with the help of inducing with 4 chemicals. Such type of stem cells are called as induced pluripotent stem cells. Pluripotency means they can be able to convert to any type of cell. So these cells are called induced pluripotent stem cells. He is the godfather of ethical concerns actually. Until we just relied on embryos, we are just killing the embryos. We are taking sperm, ova, making a small zygote and killing the zygote, extracting the cells for stem cell therapy. But internationally, there are huge, compulsions on not touch the embryos. Don't kill the embryos at any cost. See, if today we are entertaining this field to work on the embryos, silently this will become a black market. People will make, pre-mature babies or mature babies just for maintaining the market for, organ supply. So internationally, embryonic stem cell development was abandoned. There are stringent rules of not doing any research on embryonic stem cells. In that time, we got Shinny and Mack and he got Nobel Prize for this discovery, induced pluripotent. Now we can take a mature cell and we can convert it back to whatever cell we need. So that is a very big development. No more ethical concerns. Okay. Of course, there are some limitations of induced pluripotent. Some of the limitations are, see, an embryonic cells have a good renewability, but we cannot expect that renewability from an adult terminally differentiated cell. Technically, even though this became a stem cell, it don't have that agility of that L-ness is diminished in this cell actually. It will behave like a stem cell, but it is not so agile. I don't know how to tell you agility means. It is not so it don't have that replicating property. Maybe a normal embryonic cell will replicate 200 times, but this will replicate only 20 to 30 times. So because it is already an adult cell, or I can say like this, we took a old granny, we backed up her looking like a young, but internally she is already weak. So that's the concept of induced pluripotent stem cell, but better than this one. Okay? Yeah. Next, niches. I said niche. Niche means microenvironment. It is the microenvironment that is deciding the stem cell to be converted to any one of the type. There is a type of environment in the bone marrow. If we keep any stem cell in the bone marrow, it will be converted to bone cell. Either RBC, WBC, platelet or nearby osteocyte. That means bone cell. Because that is what the microenvironment guiding it. The microenvironment, there are, so the niches means microenvironment. Microenvironment has multiple factors. When I say microenvironment, it's a very basic, broad term. Microenvironment might have some chemicals, some cells, some stress in a sense, some mechanical stress or some stress. So that we'll discuss in the stem cell niches. What are the types of niches we have that guiding the stem cell to enable it to become to differentiate? Got it? So let's see what are the niches. That is another topic for you. So yeah. So these are a quick overview, whatever I discussed. So first we have embryonic stem cells, as we discussed. We are literally taking the sperm egg and we are fertilizing and we are killing the zygote. We made a zygote, we killed the zygote and we extracted the cells. Such cells are called as embryonic stem cells, but that is not ethically good. Okay. So yeah. So ethical concerns are heavy. Harvesting embryonic stem cells involves destroying embryos, leading to ethical contributions. So we should not destroy any life because already it has a life now. Yeah, that is a very challenging, ethically challenging questions. What were your thoughts on life? At what stage you will say you have a life? After birth or before birth? You got my question. Only after 9 months you are going to say you are alive or life. Where we need to define something as life? At what stage? Before birth. So let us go before birth. There are 3 trimesters. Okay. First trimester, second and third. At what stage you will, label it as a life? So up to first trimester is not life then for you. By first trimester, by first 3 months, you have a very small baby actually with all legs and hands. From second trimester onwards, the baby will grow, that's it. So where you will label something as life, that is challenging. From sperms, yeah. Sperms, technically speaking, sperm don't have a life because it don't have 46 chromosomes. It has only 23 chromosomes. So it is a half-life. So that is a boundary actually. If you take an individual sperm or ovum, that's not a life. Okay? But once you combine them to get a 46 chromosome hypercell, which has the capability to become an entire being, that is something concerning. That is when we can label it as a life. And that will be a zygote. A zygote is always a life. And we should not destroy a zygote. That's why there are ethical concerns. Got my point? Yeah. Then, next one, adult stem cells. What do you mean by adult stem cells? After birth, whatever stem cells present in our body, they are called adult stem cells. Maybe adult stem cells are multipotent stem cells. Okay. Can you give examples of adult stem cells? Stem cells in the bone marrow, stem cells in the liver. Can you guess why stem cells in liver? What is the contribution of liver stem cells? Any idea? There is only one organ in the body that can able to regrow. Even after cutting a liver, it can able to regrow itself. Because liver has an abundant amount of stem cells in its deposit. You know, they are deposited in the liver. Okay. Yeah. So yes, liver has stem cells. Any other adult stem cells? Any other locations of adult stem cells apart from bone marrow and liver? Huh? Hair cells, very good. Hair cells have skin. Skin has stem cells, very good. Then yeah, next anything? Nails. Well, when I say skin associated structures. Hair, nails and skin, epidermis. Very good. Teeth. So now, today we have a challenge. The challenge was, these teeth stems are only one time disposable. First we have baby teeth, disposed, the secondary stem cells will get activated, they make the adults, the secondary teeth. Now there is a potential research gap. When we extracted the tooth, can be able to redeposit some stem cells there, so the tooth will regrow in the adult. Is it possible? So, yeah, the challenge with such environment was, such niche was, see, in case of teeth, it's not a single entity, it has multiple lineages. Teeth must have neurons, teeth must have blood vessels, teeth must need support of bone, teeth must have support of enamel. Enamel came from mesoderm, nerves came from ectoderm, so they have various lineages. Now, is it possible to make a multilinear stem cells to work together? Then that will be a life. That is not possible. Artificially, maybe we can reproduce any one line of the stem cell, but we cannot group them together. If that is possible, we can make synthetic animals, right? So, still there is a challenges of making life. We understood stem cells are life candidates, but no. Interconnecting stem cells and communicating stem cells among themselves with multiple lineages is something very challenging. That we'll see then. So yeah, these are adult stem cells. So whatever you said is very good, adult stem cells. Now, what is your thoughts on umbilical cord stem cells? What type of stem cells are those? Are those adult stem cells or embryonic stem cells? Yeah, they used to say that umbilical cord stem cells are a type of adult stem cells that has the potency to become, a pluripotent adult stem cells. Okay, so that is one chance. Rather than killing the embryo, you can harvest embryonic cells from the umbilical cord. But that's why we have this umbilical cord G1 stem cell banks. They will harvest our cells, when during our birth, they will collect the umbilical cord cells because umbilical has the embryonic cells, okay, and they can preserve. They will preserve at -180. In the future, maybe my mom saved my stem cells 30 years ago. Now I'm 35. Oh yeah. So 30 years ago, my mother saved my stem cells in the J1 stem cell bank. Unfortunately, today I met with a bike accident and I got some abdominal fracture, some of my liver gone. Okay, so now I can access those stem cells, I can transplant those cells in my blood and they will convert my liver to a full grown liver. Or unfortunately today I got a leukemia, blood cancer. I can re-implant my own stem cells from when harvested during my birth. So yeah, it sounds good. We all can save our stem cells, right? Or at least our children's stem cells. The challenge was these banks asking around 60 to 70,000 per month to save the cells. So the cost was very high actually. Minimum 20 to 30,000 currently. So what was your opinion? Are we going to invest 30,000 just in case to use in the future? Yeah, that's one question actually.
But we can't donate stem cells doing chemo.
Donating stem cells is different. They are adult stem cells. They are not embryonic. And we need to do transplantation matching. But embryonic stem cells we need not to do matching because they are our own cells. Whereas donation, still we need to. They won't match easily. Other persons, our immune system is like
doing chemotherapy, we can't donate that.
When the patient is in chemo, you can donate to the chemo. When your blood.
We can only donate it when he is in recovery position. Not in chemo, sir, because chemo is killing the cells.
You are just mixing things. Chemo can be given to cancer, even non-cancerous patients also chemo will be given just to suppress the immune system. Especially chemo given for bone marrow transplantation. Definitely we need to destroy the pre-existing bone marrow. Why the patient went up to the chemo stage? Because his bone marrow was having issue. So yeah, so that I will discuss later. Okay. Not only every stem cell require chemo. Only after chemo will proceed. That we'll see. Any other questions? Yeah. So these are adult stem cells. Some clarity came right about adult stem cells. Then next one, whatever I given examples, blood stem cells, others, right? Yeah. Then, so types of adult stem cells we have hematopoietic, mesenchymal, epithelial. Epithelial is all skin cells. Then characteristic. Then we got this induced pluripotent stem cells. Take an adult fibroblast, take an adult skin cell. Keep in the chemical mixture. I said right, there are 4 transcription factors: C, SOX2, Nano-G, and Oct3 and 4. So keep these 4 chemicals. Adult stem cell will be converted to, induced pluripotent stem cell. This is called as reprogramming. Conversion of an adult cell to a stem cell is called as reprogramming. Reprogramming we achieved via 4 to 5 transcription factors, C, SOX2. Okay, just remember. So now, once this pluripotent stem cells they can be converted to any type of the stem cell in the body. Okay. Yes, this is about adult induced pluripotent stem cells. So this is a common table. So a tip for you when writing answers in the exam. For every answer, you should have a definition, a flow chart, a brief discussion, maybe 3 to 6 lines of discussion, then a table plus conclusion. That's it. No matter whatever question or whatever subject. Okay. See, I'm not going to read your paragraphs. I don't have time to read each and every paragraph sentence in the exam hall. I need to check 30 papers and I have very tight deadlines actually. I need to do when I say all faculties, we need to go to invigilation duty, we need to go to exam center, we need to check your paper, we need to bubble your that OMR sheet. So definitely we will be in a hurry. How you can impress the reader? How to impress the reader is my question. You can only impress the reader by breaking the topic into simplest forms. See how I'm writing on the board. This is the best way of impressing a student. Rather than writing everything, I'm just breaking down into a table. So you understood, right? So this is what I am expecting from your answers also. There should be a small definition. There should be a flow chart of the breakdown of your topic and brief discussion about your flow chart and conclusion. That's it. Flow chart with examples. If it has some examples, 20% examples of flow like this. Simple. This is the best method of writing answers. Not only by subject, any subject actually. Okay. Yeah. Why I am saying this because in my notes I have ensured to keep tables, flowcharts, diagrams and brief discussion. So please break down your answers in such fashion. Okay? Yeah. So coming to the niches. I will continue. Just 1 minute. So niches. What do we understood by niche? So the term stem cell niche refers to specialized microenvironment within the tissue that regulates stem cell behavior. Microenvironment within a tissue that will regulate stem cell behavior. Okay, including their renewability, their differentiation, and their maintenance. Okay, so there are multiple types of niches. So niches will have multiple things. These are the niches. We have some inflammatory niches, extra cellular matrix niches, physical factors, then hypoxia and metabolism and cellular. That I will discuss in the next lecture. The niches, their types and their significance, and their role and their contributions in regulating the stem cell differentiations. Okay. So thank you very much for today's class. I will take, yeah.